sequencing. Okay, so the first thing is like you would want to understand what is DNA sequencing, right? So what is the definition of it? So the DNA sequencing basically means that it is a laboratory technique used to determine the exact sequence of bases in a DNA molecule. So whenever you have a particular DNA molecule which goes on to produce a particular protein and which affects certain situation, you would want to learn the DNA molecule sequences. So that is why you use DNA sequencing, right? Okay, so let's understand what is the history behind the DNA sequencing. Okay, so the first thing is that actually what happened was that in 1955, Frederick Sanger, who was actually the first one to understand sequencing and the first one to give out a technique on sequencing, actually sequenced the whole amino acids in insulin, a small protein secreted by the pancreas. This provided something which was very interesting and which was something out of the box. Nobody thought that protein had a molecular pattern until Frederick Sanger gave it out. Everyone thought it was just a chemical suspension in a fluid. It had some chemical structure, and it was just a suspension in a fluid. That's what everyone thought. But what happened was that Frederick Sanger gave out this, and this spurred the whole community of X-ray crystallographers. So what happened? Watson, Crick, these people, they started understanding there's something connected between DNA and protein. There's something which is connecting it. So once they got to know that protein had a certain pattern, they understood ki okay, DNA is now connected to protein, and the whole idea of whatever we learn today, the most basic things, are actually a product of this particular invention. Next slide, sir. Yeah. So the basic methods of sequencing are two different. There are two different basic sequences. Sequencing in a traditional method, the two different sequencing are Sanger sequencing, which was developed by Frederick Sanger, who you see in the top right, and the other was Maxim Gilbert sequencing, and Gilbert is the one who you see in the right down. So both of them won the Nobel Prize in 1980 in chemistry for developing these sequencing methods. Yeah. So Sanger sequencing, it is one of the most classic technique used to sequence DNA. Okay. So the first thing was that you would want to know what are the items or what are the basic ingredients needed to do Sanger sequencing. The most basic items you would need is a single stranded DNA template, a DNA primer, a DNA polymerase, DNTPs and DDNTPs. I'll let you know the actions of DNTPs and DNTPs in the next slide. So can you go to the next slide and come back, sir? That was disordered, I guess. Next slide, sir. Yeah. So the process starts with denaturing of dsDNA. So you have a double-stranded DNA, but to perform sequencing, you would want a single-stranded DNA. Although today there are different methods also used, like M13 vector, which actually directly produces a single-stranded DNA. But today we are for simpler understanding, let's denature dsDNA using it, and then make multiple copies of segment. So you make multiple copies of a segment you want to study, and then attach a primer, and you take 4 different solutions in which you'll add different items, and I'll let you know what are those items. So in the four test tubes you have, in the first test tube you'll actually add, like, for all the four you add DNA polymerase, which is a must, because that will take the sequence, that will actually do the sequencing. I mean, that will actually do the building up of the whole sequence. Okay, so the next thing is that you add DNTPs to all the four equally, almost equally. And DDNTPs, you add only one type of DDNTP for each test tube. You don't add all the DDNTPs. What do you mean by DDNTP is that dideoxynucleotide triphosphate. So you will not have a 3OH in that particular molecule. So you'll have only H in that place of 3OH. So this will terminate the sequence. So after this, if you denature it, the whole thing becomes, after denaturing, you have different sequences, and then you run through electrophoresis in the 4 different lanes A, T, C, G. And when you run through it, and you can see a particular pattern, and you can read them accordingly. So that is what was shown in the earlier slide as well. In the earlier slide, what was there actually was that he instead used a modern capillary gel electrophoresis, but normally in labs today we use polyacrylamide gel electrophoresis. That's the only difference. But capillary gel electrophoresis is used in the picture which is shown right now. Next one, sir. Yeah. So, what is Maxam-Gilbert sequencing? This was sequencing which was developed after 2-3 years of the development of Sanger sequencing. So, Maxam-Gilbert sequencing requires radioactive labeling at 1,5' end of the DNA fragment to be sequenced. Okay. So, you'll have to break at certain point. You'll be degrading it directly. So what happens is that if you want to degrade purines, you would want formic acid and methylated dimethyl sulfate. Okay, so what happens, the formic acid goes and it cuts at certain positions. They are A plus G positions, so you cut them, and then after that you actually use dimethyl sulfate to cut at G positions, or you can also use pyridine to cut at like at certain positions. Okay, so they are G positions, and you have C and T, that they are pyrimidines. To cut at C plus T or C or T positions, you would want hydrazine. Now, if you add salt to it, what happens is that it inhibits the reaction of hydrazine towards thymine, and it only cleaves at cytosine. It does not cleave at thymine. So this is what it's used. But today, in the modern day sequencing, Maxam Gilbert is used very less because of the toxicity it contains. The chemicals chemicals used are a bit outrageous and very toxic in nature. So today we don't much of use Maxam Gilbert. Instead, we use Sanger sequencing only for a very small scale study or research. Next slide, sir. I'll explain the chemical. Yeah. So let me explain how it is done in laboratory. So you have four different test tubes. In the first test tube, you have A plus G, and second test tube. So how do you get A plus G in the first test tube? You increase the temperature and pH. How do you increase the pH? It's very simple, by adding NaOH, and that would cause fragments to break down. Okay, and then when you add dimethyl sulfate, that would cut at adenine and guanine positions. But now, instead after this, if you in the second test tube, if you add dimethyl sulfate and dilute HCl, it would cut at A positions only. That is at adenine positions only. And in the tube 3, the third test tube, now you would want to now you have A plus G and A. Now you would want to understand the pyrimidines part, right? So to understand the to cut the pyrimidines part, you would want hydrazine and piperidine, and that would make cuts at cytosine and thymine. Now again, as I mentioned earlier, if you add NaCl, it will inhibit the reaction towards thymine, and only C portions will be cut. So that is how Maxam Gilbert sequencing will be done. And after this, Seth, can you go to the next slide? Yeah. So after this, when you run it through gel electrophoresis, so you see when you run it through it, and now what you see is that in the first position, that is at the last position, you see that C plus T and C both are showing a positive, both are showing a positive sign towards C and C plus T is present, which means that C is present, because both C and C plus T are glowing. And next, when you see in the C plus T portion as well, you see C plus T portion glowing, but C is not glowing. That means that thymine is present and cytosine is not present. Similarly, you do the whole thing, and that is how you sequence using Maxam Gilbert sequence. So that's it from me. Thank you very much.